So thanks everyone for joining. I'm really excited to be presenting today. I'm just going to quickly go over who I am. That way, obviously, you know what I do. I'm the head of the marketing team for GenScript product division. And for today, I'm going to be talking about basically a solution that we have developed at GenScript. So a lot of you probably are very familiar with GenScript. We've been around for 20 years. We were founded as a very tiny biotech out of New Jersey. The headquarters in Piscataway, and it was actually founded with just $5,000. And since then, we've grown significantly, multiple divisions, multiple acquisitions, and obviously we're publicly traded as well. So out of those divisions, this is where the product division comes in. So a couple years ago, I joined the GenScript team, and we were kind of thinking about what is the next generation for the solutions that we wanted to develop. And one of the things that I had background in is obviously plasmid purification, but also automation. So there's a few solutions out there in the market that are very focused on mini and midi prep, but there's not much solutions that are focused on maxi or larger scale prep. So with that said, we kind of we realized that there might be a market need, and we decided that there's definitely something that might be beneficial for scientists. So of course, plasmids have been around for many, many years. Of course, basically they started to be used in a laboratory setting approximately 50 years ago or so. But the process itself, of course, is really dependent on the requirements of the plasmid. So of course, if you have research grade versus industrial grade versus the actual clinical grade, those are very different requirements for every step. For a research grade, maybe you might be working with something that, not necessarily as challenging to purify, but of course, once you get to the clinical, the requirements can change drastically, especially with regards to the purity as well as the content of what you're trying to do. So there's multiple different purification methods. Of course, each solution has its own benefits, but certainly quite a bit of them have their own challenges. So what most people are familiar with is, of course, the ion exchange, right? The column-based, the kits that are available in the market, which typically takes quite a bit of centrifugation, quite a bit of pipetting. There's multi-step processes. So what we ended up deciding to do is actually automate a solution that is based on the magnetic silica solution. So of course, whenever you're doing manual plasmid purification, you're typically doing a lot of centrifugation, a lot of filtration steps, a lot of pipetting, very labor intensive, but definitely when you get into the larger scale, when you're doing maxi and the giga and the mega preps, those end up being even more challenging because you're working with quite a bit of volume at that point. So what we ended up coming up with is an automated solution that is basically ready to go out of the box. What also was a big goal for us was to make it modular. And what that means is that you can potentially run one sample or you can run it at the maximum capacity. And the other component of the design of the system was to make it modular or scalable. So basically what you're seeing in front of you is what we call the AmMag Quatro. And the controller is the little guy that's on the right-hand side, which basically can control up to 4 purification modules. So the purification modules, each one, can run up to 6 samples at a time. So if you have a 4-module setup, you can technically do 24 samples at a time. So you don't have to run them all together. You don't have to run them with one protocol all at once or different times if you choose. And you can definitely scale it up. Let's say you want to start with six modules, so 1 module at a time, and then eventually your throughput changes and you can add multiple modules later on. So the idea is that scalable, modular, and definitely can be run separately from one another. The other key feature of the solution was to include the consumable as well as the program within this system. So that way the customer can theoretically get the solution straight out of the box, take it out, get the kit, and then run it within within a few minutes of taking it out of the box. So it is theoretically an out-of-the-box solution. Another thing, of course, to mention is that it's very reliable. The pipetting, of course, it's a pipetting module, so the pipetting is a key component. The pipetting accuracy is a CV of 3%, which is very precise. The limitation of the system is really based on the input of what you're putting into the system. So theoretically, people typically are going to run a culture volume between 50 to 200 ml. That's going to typically get you to the optimal bacterial pellet weight. So it's really, again, limited by that bacterial pellet weight. It's not really the culture volume. So it's going to be between 0.3 to about 1.5. Really, the best results are going to be kind of within that range. Typically, the runtime is very dependent on, of course, the incubations and definitely, the wash cycles that you end up programming into it. So it's going to be between 2 to 2.5 hours. And really the wonderful part about this is that you will be getting transfection grade plasmid. So there's no additional steps that you're going to have to do to get that plasmid to that pure level where you can take it and basically transfect it. And the next thing I'm going to talk about is the consumable. But before I dive into that, I did want to mention that also the user interface has been optimized and designed to really get you to that ready-to-go solution. If you want to use the protocols that are pre-programmed, you can certainly do that. However, if you choose to modify those programs, the interface is going to give you an easy-to-use way of doing that. So before I dive into the interface, I did want to talk about the consumable for a minute here. So of course, when we talk about consumable kits, if you were to purchase like a manual kit, you're usually working with kits that are available that basically contain the column and it contains the buffer. However, the solution that we came up with was actually all-inclusive. It includes basically everything that you need. It includes the tubes, it includes the tips, the buffers, even the waste. So really what you're getting is one consumable kit that's going to fit your need. There's nothing more that you really need to add. The one thing that you will need to add in complete disclosure is ethanol, because we can't really ship that, right? There's regulation around that, but that's extremely cheap. So with that said, the kit itself, what's really nice about it is that if you compare it to a manual kit, it's actually significantly cheaper. So in other words, the instrument will basically pay for itself by basically the cost savings that you will get out of the kit. So I just wanted to take you guys quickly through the process. What you're seeing in blue, like the halo box, it is completely handled by the AmMag Quatro. It's not like you're going to have to do it yourself, but I still wanted to take you through it. That way you're kind of familiar with it. So the very first thing, of course, you're going to do, which is offline, is grow your culture. And then centrifuge it down and load your bacterial pellet into the instrument. The AmMag Quatro tubes, which I mentioned earlier, they are part of the kit, and those have been designed to fit into basically in-deck vortexers or in-deck mixers that are pre-configured into the design of the system. And you will take those tubes and then you'll go ahead and load them onto the instrument. You'll go ahead and hit start, and basically, again, everything that's in blue, the halo box around what you're seeing up there, is going to be handled by the instrument. So it will go through the resuspension, the lysis, the neutralization. It's magnetic bead based, so that's the first thing that it will do to basically pre-clarify that sample. So it's going to take out that cell debris, all those contaminating proteins, the host genomic DNA, all that. And after that, it's going to go ahead and do another magnetic bead step where it's going to be binding that plasmid DNA. After that, it'll go into the washes. There's multi-step washes. Of course, those are very important to remove any kind of contaminants, as well as, of course, the endotoxins. After that, you'll go through the elution. And that last step is that where the sample is going to be transferred into a column. And that column contains a filter bed that's going to basically remove any residual endotoxins as well as potentially any carried over beads. So at that point, basically the automated process is completed. the customer or the scientist is going to take that tube off, they're going to cap it, and they're going to centrifuge it in a offline. And basically, you will get, at that point, transfection-grade plasmid. So now I'm going to be showing you the user interface. As I mentioned before, the controller, the system, it could actually control up to 4. Of course, if you have one module, then the rest would be grayed out. This is why you see there's only one with blue. And of course, when you go in, there's multiple facets that you can go into. Basically, if you want to run the program, if you want to do any kind of exports or do settings, you can do that with this interface. What's really nice about the AmMag Quatro, and I mentioned earlier, I actually worked in automation for about 15 years, so I really knew the ins and outs of what makes automation processes so important in a lab, but also the limitations of that. So what we try to do is basically make the setup almost foolproof, right? And what that means is that when the user loads the instrument, they will be able to scan the deck to confirm whether or not that the setup was accurately done. So you can see up there, there's basically a scanning feature for the tips, the reagents, the reaction tubes, the sample tubes, as well as the waste. And you can see up there, basically the entire deck is loaded. It says yes, yes, yes for every single position. Of course, if you choose not to run the full 6 sample, it will say no. You can still, of course, run it, but obviously it will give you that visual indication of what is loaded onto the deck. And of course, you can see there's actually barcodes that indicate whether or not it was loaded appropriately. This is one of my favorite features of the instrument, which is basically once you hit start, the system will give you a countdown of when the run is going to be complete. So basically, you can plan out your day, you can plan out what you're doing, and come back to the instrument. And of course, if you forget what step of the process it's on, you can actually walk up to the instrument and it will give you basically what step of the process it's on, what step it has yet to complete, which is grayed out, and then how much time is left on the instrument. So you can see it's kind of towards the right bottom corner of the screen. So now I'm going to be talking about the performance data of the Quatra. So the first case study I have, which was very straightforward, basically we ran 2 samples using the AmMag Quatro, and we ran identical samples using the manual kits that are available in the market. So what you're seeing here is basically equivalent performance. The automated solution, however, I did want to point out one thing that the team did a little bit differently than the manual kit, which is basically they ran the elution a little bit more dilute. So you can see they used a 1 ml versus a 500 UL for the manual kit. But you can see overall the performance is basically equivalent. The second case study we have was basically a side-by-side comparison to different media. So of course, there's multiple factors that impact plasmid, the actual outcome of the plasmid, right? It's going to be, definitely what kind of plasmid that you're using, if you're using low copy versus high copy, if you're using a toxic insert, definitely what kind of conditions you expose it to, and certainly the media that you use, right? The more rich the media, the more robust the growth is going to be. So here what we're basically showing is that exactly the same plasmid was ran 3 samples with LB versus TB, which of course is terrific broth. So that's a much richer media. So what you're seeing here is that for the LB samples versus the TB, we almost got almost double the outcome. So what we end up seeing also with clients, it really depends on the plasmid itself, but you can even triple the output of the plasmid if you end up switching the media. The next case study is very similar to what I had presented earlier. We basically took 6 samples. We purified them using the AmMag Quatro, and we did the same thing using a manual setup. And then what we did here is we took both those samples and we did a protein expression, exactly the same conditions for both. And what you're seeing on the right-hand side is basically the protein expression levels of the manual versus the automated. And basically, it's really equivalent in performance. And then the next case study I have was basically what we did, similar story. We took a samples that were automated using the AmMag Quatro. What you're seeing in green as well as purple were samples that we ran using the AmMag Quatro. And then what you're seeing with blue was a manually ran Plasmid. So we actually got that Plasmid from our GenScript services team. GenScript services team is really well known for Plasmid services that they provide. It is considered one of the industry standards. It's high quality. So with that said, we decided that it would be a really good control. And then what you're seeing in brown is basically just the HEC293 cells. So basically what we did here is a luciferase reporter gene. And what you're seeing is that technically what we ended up getting for the plasmids that were ran on the AmMag Quatro, again, the green and the purple, was actually better performance for the transfection efficiency versus the manual ran plasmid. So again, showing the proof of performance, but definitely the quality of the prep. And then the last case study I have was actually done for P lentiviruses. So of course, I'm pretty sure most people here know, typically when you're working with P lentiviruses, you're typically working with multiple plasmids, right? And then you're gonna take all those, probably 2 or 3, maybe even 4 plasmids, and you're basically transfecting them to create the P lentivirus. So with that said, what we did here is, what you're seeing, the table on the top is basically, again, AmMag Quatro versus the control. And basically the AmMag Quatro was purposefully dilute. So the elution that we used for the AmMag Quatro was 1.5 ml, and then for the manual, we did a 500 UL. So basically it was a 3-fold dilute sample, and again, this was done on purpose. So after that, we did the actual transfection, and we looked at the average titer for the viruses. So again, the point here was to see at the worst case scenario of the AmMag Quatro, basically a very dilute sample, versus a control sample, which is the manual prep at 500 UL, transfecting it with both and seeing how does the titer look for that P lentivirus. So what you're seeing that even though basically, again, it was 3-fold dilute, basically the transfection efficiency was equivalent. So in other words, it's really about the quality of the plasmid. And with that said, we were able to basically prove the performance even at the worst condition of a very dilute plasmid. I hope that made sense. I have actually a video that I wanted to show you, but before we play the video, I wanted to mention a couple things. So the design of the AmMag Quatro, there's a couple things that went into it. So one of the main things is that not only did we want to make the solution all-inclusive, so again, you get the instrument, you get the consumable, the programs are preloaded, but also the programs are verified, and you're ready to go, basically. But we also designed the instrument to basically get to that really optimized workflow. So there's index spinners that are custom, that had custom-made tubes that are basically coupled with those tubes that have internal baffles that enable that really good spin to both bind and clean up the plasmid. We also have puncturing tools where basically the first thing that the instrument is going to do is first scan to ensure that you have loaded it properly, but then the instrument will go ahead and puncture those cartridges, and it will basically contain all that within the instrument. So that way, not only are you getting that very pure plasmid, but also you're not potentially contaminating your plasmid prep from one sample to the next. And we can play now. Can I go to the next slide? Okay. So with that said, I just wanted to briefly mention we do have another set of solutions at the products division at GenScript, where basically it's a complete
It's really
protein.
exciting to be presenting to you.
Protein workflow. And with that said, that full protein workflow starts with the sample preparation. If you choose it, we do have an automated or semi-automated solution that can rub up to 12 samples at a time. We also have the electrophoresis or Western blotting solution that basically includes basically the precast gels, the staining solution, the transfer solution, as well as the Western. So it's a complete protein workflow. We are also working on our next generation protein purification or antibody purification system. It's going to be capable of handling up to 1 liter or maybe even more. It's still kind of in development. But the idea here is that we're constantly working towards the next solution, and we know there's certainly a market need for MIDI-scale protein purification, so that would be our next generation. And then definitely, if there's something that you guys want to see from us, if there's something that maybe you feel is an unmet need, please reach out. We're always kind of looking for what would be the next thing to do here. With that said, I did want to invite you to the booth. We are located kind of towards the lunch area in the back. It's booth number 310. We have a fun game, and you can definitely see the AmMag Quatro at the booth. And we have a lot of nice giveaways as well. And thank you. I did put a barcode up there, and if you guys did want to connect with me on LinkedIn, that's my URL code from LinkedIn. Apparently, LinkedIn can do that, in case you guys didn't know. It's kind of big. I don't know if it'll scan. So that's it, and I'm happy to take any questions.